Hi everyone, this is Sudharsanan. Today we are going to discuss about some important topics in blood physiology, commonly called hematology. In exam point of view, there are 5 major topics which includes erythropoiesis, blood grouping. Among blood grouping, ABO blood grouping is most important, and immunity. More specifically, cell-mediated immunity is asked frequently, so we have to discuss it more clearly. And anemia. Anemia in each type is very important. And finally, polycythemia. Today we are going to discuss erythropoiesis in detail. Alright, let's get into the video. Today we are going to discuss about erythropoiesis in detail. What's the outline? First of all, we are going to discuss the intro. What is erythropoiesis and its definitions, iron absorption, vitamin B12 absorption, role of kidney in erythropoiesis. What are the stages of erythropoiesis? Let's see about the definition of erythropoiesis. What is erythropoiesis? Let's pick the term. Erythropoiesis. As you all know that erythro means red blood cells. Poiesis is a Greek term which defined to form, which means RBC formations. Where it forms, RBC is generally formed in bone marrow, specifically red bone marrow cells. What is bone marrow? Bone marrow is a spongy tissue. It is soft spongy tissue present inside the bone, especially in hip bone, thigh bone, like that. Among the bone marrow, it is of two types: red bone marrow and yellow bone marrow. Where the red blood cells are produced are red bone marrow. Yellow bone marrow is basically it's composed of fat adipose tissue, so it is called yellow bone marrow. Okay, what is the major causes of our erythropoiesis? What drives the erythropoiesis to taken? The most important, there are more physiological and pathological conditions. The most important condition is hypoxia. Inadequate supply of oxygen to the tissue. The term is hypoxia. In hypoxic conditions, erythropoiesis has to be taken more. And in pregnancy, erythropoiesis occurs more. Physical activity. After the physical activity, EPO formation. If erythropoietin formation, hormone formation more, then erythropoiesis occurs. Some hormonal controls or some hormonal factors. These are the conditions where erythropoiesis occurs. Okay, let's see about iron absorption. Our body needs some iron, minerals, some vitamins to occur erythropoiesis. First of all, we discuss about iron absorption. In our diet, we take iron, which is in Fe3 plus form. The Fe3 plus form is get to the stomach. After stomach, we go to the intestine, specifically duodenum. Let's get a cross section of the duodenal cells and zoom it out. This is the duodenal cells. This is the duodenal cells. Let's right here. In diet, iron is in Fe3 plus form. Okay. After reaching the duodenum, Fe3 plus is converted into Fe2 plus. Fe2 plus is the absorbed form of iron. For this transfer, we need a iron reductase enzyme. That reductase enzyme is present in the duodenal cells, which is named. It is a duodenal cell, right? Then duodenal cytochrome B type 2 enzyme, which is the reductase enzyme. So it converts Fe3 plus into the Fu2 plus. Okay. then it get into the cell, right? Then Fe2 plus is going into the cells, but Fe2 plus going along with the H plus only. So H plus also get entered, the Fe2 plus also get entered. Okay. There is a specific transporters which helps in the transfer of Fe2 plus from outside to the inside the cells. The specific transporters present in the diurnal cell is named as divalent metal transporter, DMT. Divalent metal transporter. Okay. Now Fe2 plus we take in the diet is enter the stomach and coming to duodenum. In duodenal cells there is enzymes which convert Fe2 plus to Fe2 plus, which get absorbed into the duodenal cells with the help of the divalent metal transporter. Now Fe2 plus get inside the cell, right? Fe2 plus is not supposed to be in the free form. If it is in free form, then it leads to the accumulation of more free radicals. It is not good for our health. Okay, then it gets more damage to our tissues. So Fe2 plus must be attached into some other any tissues or forms. Like here, Fe2 plus is get attached to the enzyme named apoferritin protein. Let's use black color. It's not apoferritin. The entered free form Fe2 plus is get attached to the apoferritin. Okay. Fe2 plus. This apoferritin along with Fe2 plus is converted into, that is term, ferritin. The free form Fe2 plus get attached with apoferritin and turn into ferritin. This ferritin accumulates more and more and more, then becomes homosiderin. Homosiderin is the temporary storage form of iron. All other ferritin are not turn into homosiderin or not accumulate into homosiderin. Some of them are get released into the bloodstreams, right? Then ferritin is come out of these duodenal cells and get entered into the bloodstream. Now this ferritin is come out of the cells. Some protein is used to get out of the cells, which is called commonly called ferroportin. Ferroportin helps to transfer the ferritin from inside the duodenal cells and to outside. Ferritin is now present in Fe2 plus form, right? Because it get converted into Fe3 plus is converted into Fe2 plus and get outside as Fe2 plus only. Now this Fe2 plus is converted into Fe3 plus with the help of the protein called hephacetin, which is present in the membrane of the duodenal cells. It is Fe2 plus is converted into Fe3 plus form with the help of hephacetin. Okay. Here Fe3 plus is converted into Fe2 plus. Here Fe2 plus is converted into Fe3 plus. So it is commonly what happens? Reduction happens. So iron reductase enzyme. It is commonly iron oxidase enzyme. Hephacetin, iron oxidase activity. Okay, it gets lights. Don't bother with it. Okay. Now we got Fe3 plus in outside the cells. It get entered into the bloodstream. As we already discussed, Fe2 plus or Fe3 plus, whatever it is not supposed to be allowed to in free form. So it get attached to the transferring in the bloodstreams. transferring. Okay. Now Fe3 bases get attached with the transferring. Okay. This is the ion absorption generally take place. Let's quickly recap. In diet, we take Fe3 plus form of ion. It get attached. It get into the stomach, then goes to the duodenum. In duodenum there is a specific transporter and specific enzymes which converts Fe3 plus into the Fe2 plus. Since reduction happens, this helps in ion detection enzyme. This Fe2 plus is get converted. After converted into Fe2 plus, this get inside the cell with the help of the divalent metal transporter. Fe2 plus is not supposed to be in the free form, so it get attached to the apoferritin, then converted into ferritin. The ferritins are accumulated into form homosiderin. All the ferritins not accumulated. Some of the ferritins transport out of the cells with the help of the ferroportin. Fe2 plus form is only get outside the cells. So Fe2 plus is converted into the Fe3 plus oxidase enzyme activity. So there is ion oxidase activity which is done by efacitin enzyme. This is Fe3 plus is get converted into the not get converted. Fe3 plus goes into the bloodstream and then attached with the transferrin. Okay. Not only in diet, not only we get Fe3 plus, right? We also take some carbs, amino acids, proteins, not proteins, actually amino acids are accumulated from proteins, fatty acids. These carbs, amino acids, fatty acids are enter into the stomach and get converted, not get converted, actually enter the bloodstream. These carbs, amino acids, fatty acids are helpful in stages of erythropoiesis. Let's discuss later. Okay, now B12 absorption. We finished in intro ion absorption. Let's get this B12 absorption. Actually, before B12 absorption, folic acid. Folic acid is also important in the erythropoiesis. In diet, we take folic acid also. Folic acid also get into stomach and duodenum and enter the bloodstream, and which helps in the erythropoiesis. Okay. Vitamin B12. Vitamin B12 plays a major role in erythropoiesis. In diet, we take vitamin B12 which enters the stomach. Here, in stomach there is a cell called parietal cells. As you all know that parietal cells, there is some specific function for parietal cells which produces the factor which helps in the absorption of vitamin B12. The factor is termed as intrinsic factor produced by parietal cells which helps in the absorption of vitamin B12. The factor is called vitamin intrinsic factor. Vitamin B12 get attached to this vitamin B12 get attached to this intrinsic factor and got absorbed. Okay. Intrinsic factor and vitamin B12. Now we enter into the ileum. Okay. This is the ileum cells. Vitamin B12 is combined with the intrinsic factor and enter into the ileum, where receptor mediated endocytosis occurs. What is receptor mediated endocytosis? Receptor mediated endocytosis. Intrinsic factor is combined with vitamin B12 and get absorbed. This get into the ileum cells. In ileum, there is a special process take place, receptor mediated endocytosis. With the help of receptor mediated endocytosis, this vitamin B12 along with the intrinsic factor get into the ileum and transport into the bloodstreams. Get into the ileum with the help of receptor mediated inside cytosis inside the cytoplasm. Receptor mediated endocytosis. Now it get into the ileum cells. Then enter the bloodstreams. Where vitamin B12 got separated. Before entering the bloodstream, vitamin B12 got separated and intrinsic factor got separated. Where vitamin B12 only enter into the bloodstream. Actually, vitamin B12 enter the bloodstream. This is bloodstream. In bloodstream, vitamin B12 get attached to the transcobalamin. Transcobalamin. In bloodstream, vitamin B12 get attached to the transcobalamin. This is the absorption of vitamin B12. What is the role of kidney in erythropoiesis? Kidney play a major role in erythropoiesis. This is a structural kidney. There is a millions and trillions of nephrons present in the kidney, as you all know that. In nephrons, there is a proximal convoluted tubule. The PCT, proximal convoluted tubule, there is a factor which is called hypoxia induced factor. Hypoxic induced factor. Hypoxia inducible factor. Whenever there is a hypoxic condition in kidney, proximal convoluted there is a HIF. When there is a hypoxic condition, it got induced and promotes transcription, and after translation a protein is formed which forms the important hormone called erythropoietin. It is a super important hormone which helps in erythropoiesis, named erythropoietin. This is very important. Erythropoietin hormone is helpful in erythropoiesis. Okay, the kidneys role in erythropoiesis finished. Let's discuss about the stages of erythropoiesis. Stages. Okay. First of all, there is a stem cells called erythrocytoblast. It is also called hematopoietic stem cells. Erythrocytoblast, it is a stem cells or hematopoietic stem cells which gets converted into myeloid stem cells. And lymphoid stem cells. This lymphoid stem cells is converted into B cells, T cells and mast cells. So no role in erythropoiesis. Let us focus on myeloid stem cells. Myeloid stem cells is got differentiated into the proerythroblast. The hormone produced here, erythropoietin, which helps in transformation of myeloid stem cells into proerythroblast. This induces this process. Proerythroblast actually large. It contains nucleus. Nucleus is prominently visible. Normally mature RBCs lack nucleus, right? Proerythroblast contains nucleus and nucleus is more prominent. After the proerythroblast, the next stage is early erythroblast or early normoblast. It is also called as basophilic erythroblast. Nucleus is large and prominent in proerythroblast. Now nucleus become smaller because it progressively going to degenerate and lack of nucleus in matured RBC, right? Reticulocyte cells also lack RBC only. Nucleus. Early erythroblast is also called as basophilic erythroblast. Nucleus is small and cytoplasm is darkly stained, basophilic stain. After the early erythroblast, there is a next stage called polychromatic erythroblast. Polychromatic erythroblast. The term itself says that polychromatic, mixing of both blue and pink color. Actually, okay, ribosome becomes blue stain and cytoplasts are pink stain. So there is a mixing of both blue and pink stain. So it's named polychromatic erythroblast. Polychromatic erythroblast is transferred into orthochromatic erythroblast. Orthochromatic erythroblast. This orthochromatic erythroblast is pink in color. Nucleus is almost deeply condensed. Here, prominent nucleus, then progressively degenerate. Here, small and small. It is almost condensed. In reticulocytes, nucleus is completely absent. Nucleus is completely expelled out. The next stage is called reticulocyte. After the nucleus completely expelled, the cells which enters the circulation is termed as reticulocyte cells. This reticulocyte cells is converted into the mature RBC. Mature RBC, which is commonly called erythroblast. Which completely lack nucleus. All the stage occurs in 7 days. Okay. That's all about erythropoiesis. Let us quickly recap. We all know the definition: formation of erythroblast, formation of RBC in red bone marrow cells. Major causes hypoxic condition. Hypoxic condition is the most major causes. Iron absorption in diet, we take Fe3 plus, which goes into the stomach and duodenum. In duodenum, that converted into Fe2 plus, and Fe2 plus along with the H plus with the help of divalent metal transporter get enter inside the duodenal cells. Fe2 plus is not supposed to be in free form, so it get attached to the apoferritin, then converted into ferritin. The group of ferritin is formed into the temporary storage form called homosiderin. This ferritin is again come out of the cells with the help of ferroportin. Then Fe2 plus is converted into Fe3 plus. Iron oxidase activity is happened here with the help of epacitin. This Fe3 plus entered into the bloodstream and combined with the transferrin. This is all about iron absorption. B12 like that, carbs, amino acids, folic acids and fatty acids are all enter the stomach and enters the bloodstream, which are helping the erythropoiesis stage. Vitamin B12 is not simply absorbed in alone, so it gets acted as an intrinsic factor which is produced by the parietal cells in the stomach, which get converted into not get converted, it combined with the vitamin B12, then enter the ileum. The receptor mediated endocytosis helps here, and vitamin B12 enters the bloodstream and combined with the transcobalamin. Role of kidney. Kidney play a major role in proximal convoluted tubule. There is a HIF, hypoxia inducible factor, which got induced whenever there is a hypoxic condition, which produced the important hormone erythropoietin, which plays a major role in erythropoiesis stage. Erythropoiesis stage is normally erythrocytoblast stem cells is differentiated into myeloid stem cells and lymphoid stem cells. Lymphoid stem cells here is not concerned more. So myeloid stem cell is converted into proerythroblast, which contains a large nucleus, and nucleus is prominently visible. Then it converts to early erythroblast, which is also called as basophilic erythroblast. Small nucleus, cytoplasm is densely stained. Then it converted into polychromatic erythroblast, mixing of colors. Blue color ribosomes and pink color cytoplasm are seen. Then arthrochromatic erythroblast are seen that pink color spells nucleus is condensed more. Then reticulocyte cells. After nucleus is completely expelled, the cells which enters the circulation is termed as reticulocyte cells. Then it can convert into the mature RBC with completely lack nucleus. Okay, this is all about the erythropoiesis.